Good morning, students. Please put your attendance. Okay. Today we'll be continuing our discussion on the industrially relevant microorganism screening. In our last class, we talked about continuous bioreactor operation, CSTR operation, how is it can be useful for isolation of industrially relevant microorganisms. I will be sharing some portion from your textbook as well as PPT in this class. Okay. So when we are talking about this selection process, there could be some reason and some limitations also. We'll be discussing on that. So when you are talking about, let me see, yes, the continuous culture, what is the limitations there? So first we'll be talking about limitation of continuous culture. I hope you understand what is the purpose. Purpose is that in a continuous tank, stirred tank, there is different kind of microorganism. As I was telling, I'm just repeating for just few minutes so that you will remember the things. And I'm having some other microorganisms. You want to select this one, for example, and you don't want this microorganism to survive. So there is a choice you have, and you must know what is their specific growth rate. Specific growth rate for microorganism B, specific growth rate for microorganism A. This procedure is only useful if I know that your microorganism specific growth rate, what you want to isolate, is greater than other organism, that is others. That means your microorganism is fast growing. You try to understand. Your microorganism is growing faster than the other competitor. So what will happen? There is a continuous feed, as I told you in our last class, if you remember. If you continuously provide input of substrate and continuously it is going out, what will happen? If I increase the flow rate, this highly growing microorganism can still be there because there is always some loss, washing out. If this is growing faster than this one, I can reach some time where the dilution rate, whatever I am providing to this reactor, is going to be bigger than growth rate of this microorganism. So if I provide high dilution, okay, what is dilution rate? By the way, if I say dilution rate, so if dilution rate is greater than any organisms B, then the B organism will be removed. What does it mean? It means that, what is dilution rate? First of all, you see, the tank volume is, for example, V, and flow rate is, let me say, for example, I am having a tank. Please listen this example carefully. I am having a tank of some 10 liter, and the feed you are providing is 2 liter per minute. Okay, tank size is 10 liter and the food is coming is 2 liter per minute flow rate. So there is one terminology possibly you'll be studying in your upcoming class, that is called hydraulic retention time. That is volume, how much we are having by flow rate. That means 10 liter by 2 liter per minute, that equal to 5 minutes. You see, you do the calculation, it will be 5 minutes. What does it mean? It means that on average, anything coming in the feed, any molecule is coming, it will have 5 minute time, it will stay here, then go away. Very simple. If I'm putting some, for example, this is normally water is coming, and if I'm putting just one molecule of sugar, and I assume that it is not broken down, then I can assume that sugar will stay here, or maybe some small dust particle, for understanding. If I put something here, it will stay here normally about 5 minutes. After that, it will go away, because the tank size is 10 liter. So 5 times 5 minutes, how much material is coming? It will go away after 5 minutes. That means 5 minutes is the time someone is going the opportunity to stay here. That is called HRT. And dilution rate is just opposite, inverse of HRT. That means 1 by 5 per minute. Inverse, actually. 1 by, you see, 1 by 5 minutes. That means 5 minutes will be inverse. So this is called dilution rate. You can calculate. Similarly, microorganism growth rate you also know. So for example, I am having a microorganism B, which growth rate is, for example, 0.1 per minute, for example. And here dilution rate is becoming how much? 0.2 per minute. What will happen if I make a flow rate up like this? After some time, as I told you, if the dilution rate becomes more than microorganism specific growth rate, it can be eliminated. So mu B will be gone. After some time, there will be no mu B, only mu A will survive. So this is one of the approach for screening, but this is only useful if you are going to isolate a microorganism which is having higher growth rate than all other competitive microorganisms. But that is not a limitation. I started the discussion on limitation. The first thing is that establishing an steady state is not easy. As I am telling so easily and smoothly that I will maintain some constant flow rate, there will be constant growth rate. Not like that. You will always have some transient increase or decrease in the biomass. You cannot really establish a constant density of biomass in the reactor. So this could be one of the important problem where the continuous continuous cultivation process for isolation is a challenging. That otherwise this could be one of the important aspects. Now, when we are going for enrichment, other methods are also there. I already told you. What are the other strategy that I'm going to discuss now? So first strategy is that, okay, we are looking for some, I told you, one enrichment can be made by substrate. Okay, substrate feeding. That means you are allowing the microorganism only to survive in your plate. What is the example? I am preparing a media only containing starch. So only the enzymes which can hydrolyze starch, okay, amylase enzyme, for example, I'm having. If the microorganism is producing amylase and we are growing on a plate or liquid culture which containing the starch, they will only survive. Other people will not survive. This is one approach. So you are actually restricting the feeding compositions or substrate compositions, whereby you can actually manipulate the procedure. You can actually isolate some specific microorganism depending on substrate. Another way also people can also do is going for inhibition product, for example. What could be the possibility of inhibiting? Let me talk about one microorganism. This microorganism, for example, taking your substrate or food and producing something. What does it mean? I know the if you are studying biochemistry nowadays, you might be knowing that when they are taking sugar metabolism, do you have biochemistry this semester?
Yes, sir.
Okay, so I think you are already studying this glycolysis, Krebs cycle, all those sugar metabolism pathways. And if it is true, then you must be knowing that there is always in the glycolysis, for example, there is producing pyruvate, and in Krebs cycle, tricarbolic acid, TCA cycle. Those are the final end products. And if it is aerobic process or anaerobic process, depending on the process, we can also have accumulation of different acids, lactic acid, or we can have ethanol if it is anaerobic process. So basically, depending on, of course, the metabolism will vary. So basically, from some substrate, there is some cycle is going on, metabolism is going on, and some end product is coming up. Now, what happens? If I put more end product here, that means, for example, the microorganism is producing lactic acid, for example. It is product. So it is taking the sugar, and finally, after all those metabolism, it is producing lactic acid. That means lactic acid is the end product of the process, right? But if I'm preparing a media already high concentration of lactic acid, if I am already providing there, okay, it may happen that lactic acid is inhibiting the growth metabolism, because that this is called product inhibition. And in that case, what will happen? So for example, let me speak very clear way. So for example, I'm preparing one media which contains high concentration of lactate. Lactate is, or lactic acid is, for example, our end product. And in this case, microorganism, if there is already some microorganism which can also produce lactic acid, they will not be happy, because if there is a high concentration of your end product, what it is producing itself, if it is already there, high level, then there will be causing an inhibition. So this can be causing as product inhibition. Probably you will be studying those things more in your enzyme technology classes if you are having. So product inhibition can be one of the strategy where we can block the production of certain, I mean to say, high concentration of the product I can provide, which will block the metabolism of the microorganism. And the microorganism which only depends on the pathway, they have no strategy to survive, they will go away. So this could be one of the important selection criteria. One can be substrate, one can be this one, and third one is, of course, inhibitor. Because what are the inhibitor? So as I am telling, some substrate it is taking, it is making some intermediate things, I1, I2, like you know, right? And finally, it is going to have some product. These are all carried out by some enzymes, right? You know all the enzyme system when you are studying your glycolysis and Krebs cycles. There is all enzyme is involved. By the way, the enzyme is not only specific for your substrate. Some of the enzyme can bind with other chemical moieties. Other chemicals also can bind. For example, I'm putting some urease inhibitor. So these inhibitors will actually behave like the substrate itself. So what will happen? This enzyme, instead of working on your intermediate, they are going binding with this one. And finally, there can be some competitive inhibition or non-competitive inhibition. That means enzyme is mostly used up by false chemicals. That means in the media, I am putting some inhibiting agents. The inhibitor role is that they are very similar to your intermediate substrate structurally, okay, not functionally, structurally similar. So what will happen? These enzymes, as I am talking about enzyme 1 or enzyme 2, they can come and they can bind with your chemical what you are providing. So what will happen? The actual worker who are actually doing, the enzymes are actually worker, right? They are actually converting the chemical reactions, intermediate. So if the enzyme is all utilized in these non-useful process, enzyme will not be available for conversion of IT to product. What is the result? Result is product is not being formed. That means microorganism, this process, if I stop something in the intermediate process, the earlier steps will also be blocked, like it is a water flow. If I put a barrier here, so it will stop the all upcoming water also, it cannot come, so it is stall. So similarly, if I stall this process, earlier process will also be, and that is called feedback inhibition. Those terminology you will learn later, not now. So this inhibition is actually going to make no use of this particular microorganism to use that particular substrate. This is a failure. So this failure means these microorganisms can be easily removed. So you see, isolation means what? Isolation does not mean that we will only select something. Isolation means that we will remove the others also. Anyway, for example, as I am telling this example, I am going to isolate one specific microorganism which is utilizing citrate. That means I am going to, if I can do that by simple two strategy, either I am selecting the citrate utilizer or I am inhibiting those one who are not utilizing citrate. So both will do the job. Here we are talking about, in this slides we are talking about inhibition of those competitors who are going to utilize that. So basically we are reaching in the same goal. Okay, this procedures is really uniform and unified, and we can have the same process for multiple reactions. Now that is for few cases. We can also have some situations here. We can also have some situations this is more complex actually, that you do not have any specific substrate or inhibitor. What does it mean? I mean to say some substrates are so common. Okay, I told you about starch. Okay, good. Starch you can use when I'm going to isolate starch degrading microorganism. But for example, I am going to isolate one microorganism which is going to produce some amino acid synthesis, or I'm going to look for a microorganism which is producing intermediates. What is the intermediates? For example, your TCA cycle intermediates I'm going to look for. What will happen? In that case, our substrate can be very simple sugar. For example, I'm using glucose, and most of the microorganism A, B, C, everyone will use that. So you cannot really control that whether A will grow or B will grow or C will grow. Of course, in starch you can have the opportunity, right? If I'm putting starch, only some microorganism which is breaking down the starch, having for example amylase, they can break down. But here if I'm using some sugar and our desired product is not very specific for any particular group of microorganism, what you will do? Then this becomes a challenge. In that case, you cannot use these kinds of approach. What we will use? We will use molecular biology approach. How does it mean? Okay, first of all, let me talk about how the molecular biology tools are going to help you. any property, any pathway, any metabolism, probably you are now studying, any protein is produced, any RNA is produced, nucleic acid production, everything is coded in your gene. I mean to say the microbial gene, right? So whole DNA, some segments, this, for example, this section of DNA is producing one of the enzyme. This section are producing some another enzyme. Their enzymes may be of different functions, right? So this is one gene, this is another gene, this is also another gene. Maybe some of the genes are producing proteins, different kind of proteins. Some of the genes are involved for protein synthesis machineries, maybe some RNA synthesizing genes, different kinds of RNA, that is molecular biology aspect, I'm not talking. And there is also a gene which can actually utilize, for example, some microorganism is producing some polymeric substrate, and we know this gene is involved there. So if I know the gene already, I should also know the gene sequence. I mean to say, you should know, if I'm saying 5 prime, 3 prime, what is the composition, AT, G, C, G, C. So this gene sequence is available in different database. You can always look for example, I know this is the gene sequence from NCBI database. I'm looking for some product. I mean to say, I know the gene which is involved for production of certain metabolites. So if I know the gene sequence, I can actually, I'm having these A, B, and C different clones, right? For example, I'm isolating some microorganism from soil and I plated them. Okay, now what can I do? The whole, for example, I'm having the whole colonies. Each colony, actually, I can separate it out. I can take out each colony and I can grow them separately. And then what I will do, I'll take out their DNA. Okay, for example, I'm breaking the cell. DNA isolation steps is not part of this class. But anyway, you can break down these cells and you can isolate the DNA. Once your DNA is available, and I know this gene is likely to be there in your target. For example, this microorganism is producing some amino acid, and you know the amino acid synthase gene is this sequence. So I can put this sequence some primers. Primers means actually they are going to bind. And if this gene is present here, I can run some PCR and I can amplify. Those terminology, I think you don't know right now. Or maybe you know not very clearly. So basically, in summary, what I'm trying to say, there are many metabolites, there are many pathways, there are many products which you cannot control by your simple strategy, putting some substrate, inhibitor, temperature. No, it does not work some cases. In that case, what you will do? You have to grow this culture, you have to make them isolations of these microorganisms on plate. Everyone will grow. I cannot stop A, B, C, D, everyone is growing here. Then what I am doing, I am individually growing them, each colony separately, separate pure culture. And then I will try to isolate their DNA. And now I know if my product is being produced by this microorganism, this should be its primer sequence. I know. That means what will happen? If this specific sequence is there, I can actually amplify. I can increase the number and I can go through PCR and I can identify, yes, this is there. We can also have the opportunity to identify specific microorganism by 16S rRNA. You know that? That is for molecular identifications. What does it mean? What I'm trying to say? Let me come into one important aspect. So you are going for isolation of microorganism for a specific property, and you, for example, it is degrading some genobiotics. It is degrading phenols, and I know that Pseudomonas species are all known very good degrader of toxic compounds, toxic metabolites, for example. That means you know the genera, right? So what will happen? What is your expectation? If I know the whole microbial kingdom, there will be different class of microorganism involved in different metabolic activities. And if I know that Pseudomonas, for example, one of the very important group of microbial species, which can have the potential to degrade different xenobiotics. So what happens? All Pseudomonas, for example, Pseudomonas flavans, Pseudomonas fluorescens, Pseudomonas putida, these are all coming under genera of Pseudomonas. Their most of the functions are very similar. And what happens? In their genome, some portions are conserved, very similar. Some portion, that is 16S Rrna. Some portion will be conserved for a genera. Some portion will vary because species is different, right? So what will happen? If you are looking for a property, first of all, I am having some soil sample and I can take out the whole DNA fragments and I will look for whether there is anyone matching with this sequence of Pseudomonas species. So if we are running some 16S rRNA analysis, we can also identify whether this particular genera or species is there. Okay, you can study more on this on 16S rRNA analysis. I can tell you, if you give me some microorganism, I can tell you what is its genera, what is its species, without doing anything, only by molecular techniques. So once I know the molecular technique, I can find out, I can limit the possibility. Yes, I know that Pseudomonas species is very good, well known, and then I will look into it. That means you are actually screening not by its physiochemical properties, by based on its genomic data or molecular biology data, whatever you are having at these positions. I'll come to this point using your textbook now, because I need to have some examples specifically. Let me see. I hope you are understanding some of the concepts, right? Everything may not be immediately understandable because you have not gone through these molecular biology techniques, at least much of them. But basic concepts should be similar. Now coming into your textbook here, when I am talking about inoculum development process, I already talked about that in our last class. What should be, not this one, I'm sorry. This is inoculum development. Here, isolation and preservation, isolation part more importantly. This aspect I have already talked, what should be the ideal temperature, what should be the suitability there of the microorganism, and how they, sorry, I'll reduce the font, and how we are doing it. So first one, a possibility is ideal isolation procedure. I already talked in our last class using soil. We can put some selection pressure on that. I already told using substrate, that is yesterday's discussion. I think we are already talking about, yeah, that is also one of the approach. You see, as pointed out, for screening program to continue generate new product, it is becoming increasingly more important to concentrate on lesser known microbial taxa or utilize specific screening test. So we need to know more test on that, because from taxonomical database, if you look for the taxonomical database, you can also, taxonomical database means I need to know which genera is known to produce that, which genera is non-producing that. In our last class, we already talked these aspects, that is continuous enrichment culture. Sec flask, it is not possible, but I think we can do this if I know the specific growth rate aspects here. You see, I already told the prevalence of organism in a batch enrichment culture will depends on maximum specific growth rate compared to the maximum specific growth rate of other microorganism. So it will be only possible if the fastest growing microorganism will be actually surviving. And we can do this using continuous or chemostat culture. I think we are talking it here. Continuous enrichment technique we talked. Please go through this. You see, the enrichment process should be designed such a way that predicted isolate meets as many as criteria proposed for possible successful isolation of the organism and production of biomass. So we must need to know. Inclusion of other organic carbon sources may be also included. The main difficulty in, okay, this is the difficulty. The main difficulty in using the continuous enrichment culture process is the wash out of the inoculum before an adapted culture is established. I was telling that I will do some flow rate, but it may happen that they never reach the steady state condition. If they don't reach steady state condition, before you are starting even, your desired microorganism can also go away. So you must be careful how much microorganism, how much inoculum you should have. If it is very less amount, then your desired microorganism will also go there. And how can we avoid that? The first, use a turbidostat, second, use a two-stage chemostat. So I can stop this by using measuring the biomass. Turbidostat means, what is turbidity? That means I am maintaining a constant turbidity. If the turbidity is very low, what is turbidity? If there is more microorganism, the media will be more turbid. So I am also putting one controller in bioreactor, where they will not allow it to go down below certain level. That means someone must survive, right? Otherwise it will not continue to flow. Flow will be stopped. So turbidostat can be one of the approach. And two stages, first stage, you see, two stage I should talk. I have not talked that. Two stage chemostat. Okay, what was the problem in one stage? Only one problem. I was telling that there are two microorganisms A and B. I will have a flow rate so that B will go. But it may happen that some B is going, but A is also going away more. So that means here A is not protected. A is also going out, B is also going out. Then what can I do? I can have another big, maybe possibly larger size fermenter, volume is a little bit bigger, and I am having two stage. So I am connecting this reactor, output of this reactor also together. So what will happen if I am having two reactors in series or in sequence? It may happen, for example, here A and B ratio was, let me talk about 50-50 initially, and then I am maintaining a flow rate so that A become 90% here and B become 10%. That means B is still there, population ratio I am talking. But when I am having another reactor, I will change the flow rate or volume, I will increase so much so that if the volume is increased more, what will happen? The residence time, they will get more time to stay. That means normally you see B is nearly being cleaned up, but as I am allowing here more time, A can have more proliferation and B will also have, but little bit lesser. That means I am increasing the ratio of A and B further. That means, for example, I am now making 99:1 possibility. What does it mean? It means I am increasing the possibility of recovering the desired microorganism. I told here that increasing the reactor volume. It is not always true. If I am having the opposite picture is required, I need more allowance for B and less allowance for A. My reactor can be short one also, small one also. It is possible. It is not that always bigger. Bigger is better, not like that. Depending on what is the growth rate we are giving and whom I want to allow more time or less time. More time means I will have more probability of the high biomass will increase. Less time means if I am not allowing much time, that means B will be quickly eliminated. I will have still some amount of biomass of A. So flow rate can actually, flow rate and volume combinations by two CSTR in series can be adapted. So this is one of the simple strategy people can follow when they are going to isolate, but only for industrial process people do, otherwise you do not really do. So turbidostat can actually determine the cell turbidity and they will be maintaining. The use of turbidostat will result in selection on the basis of maximum specific growth rate and it's operate at high level of limiting substrate. This is one approach we told. And when I am using two stage, first stage of the system is used to continuous inoculum. For the second stage constitute of large bottle. I'm telling na, large bottle containing the basic medium inoculated with the soil infusion. That means I'm providing two different feeds. Continuous inoculation can be employed, increasing the absorbance was absorbed in the second stage. So I will continuously improve the growth until the cell you require is going to increase. Okay. So this process has been always used and has been mostly effective. But here, the next one, what I am going to say, this one is for solid. Solid is not much control. If I am growing solid media, the most of the time the controlling factor will be pH. No other factor can really control or really manipulate the procedure more effectively in a solid state fermentation. Solid state growth will not be as high. You cannot have such much control. So here actually involve the use of selective medium incorporating substrate of the enzyme which encourage the growth of producing type. What kind of microorganism can be used? These kinds of selection mostly comes from utilizing solid state fermentation, where your media is solid and your enzyme production will actually liquefy the substrate. Enzymes, pectinase, cellulase producing enzyme, lipase producing enzyme can be some of the substrate, amylase producing enzyme can be used. So these enzymes actually break down solid substrate mostly and liquefy them for food. So we can use those substrates in solid state fermentation, and we can also have some pH adjustment onto that, so that it will be, for example, here the soil sample were pasteurized and eliminated the non-sporulating organism, then spread into surface of agar medium pH 9 to 10 containing dispersion of insoluble protein. You see, that means I am looking for some protease. Okay, if I'm going for looking for enzyme for protease production, you see, alkaline protease. We have done this work earlier. Alkaline protease will be produced at alkaline pH, and protease job is to break down protein into small peptides. These peptides will be food for the microorganism. So if I'm using some insoluble protein, what will happen? They can be broken down by the microorganism and you can select it. So they will find some clear zone also if it is insoluble protein is there. So if I'm having a solid media containing insoluble protein, I'm going to add the microorganism. The microorganism will release the enzymes. Enzyme will break down the protein. They will grow. And what will happen? If they're breaking down protein, they will make this zone very clear. So clear zone you can find out. Here, isolation method, this strategy I was talking today, isolation method not utilizing selection of the desired characteristics. So we are not going to have very specific control there. The synthesis of some product does not give the producing organism any selective advantage. I told you, right? Something it is producing does not make it something important always. It is no advantage. No advantage means then you cannot control. What example advantage? Cellulase enzyme, it is advantage. Okay. But if it is a protein, some proteins or amino acid it is producing, no much advantage over there. So in these cases, it will be a problem. I need to do something more important selection strategy should be there. And how can you do that? I told you, you need to know some unusual taxa. You should isolation process you should be doing. Some unusual taxa which is not well known earlier, that can be exploited, that can be used. Okay. And for example, we can use some taxonomical database to look for the selective for particular taxa. I was telling that you must look for database. Database now available is NCBI database. PubMed is having the literature on those databases. So you can go and you can check, I'm looking for some property, some chemical synthesizing property, then I can have for PubChem. I can also look for PubMed and NCBI to look for their genetic characteristics, their, protein, whatever they are synthesizing, their gene sequence you can find out. So basically you have a lots of data available already, and you can look into that to look for any specific property, specific products generated by this particular taxon. Incorporation, for example, incorporation of particular antibiotics into isolation media may result in selection of some resistance taxa also. So this, anyway, this is not actually usable to us, because most of the time they will be looking for someone producing some antibiotics. So this can be used to optimize the isolation of media when I'm looking into bioprocess media developments. Okay, so here you can see we are, for example, I'm going to look for gentamicin. So micromonospora we know. So we have some selective media where we can put some primary components for selection, and that can also, looking into the idea, you can also go in this similar approach. Similar approach can be taken. You know the genera, you know the species, you look into the database, you then fix what can be the possible target, what could be the possible selection pressure if you can put, and you can achieve some of the selection from natural resources. So until this, we are actually talking today. We'll be talking on screening methods, more details, how they are going to go for antimicrobial properties, beta lactamase genes. But these stories may be always a little bit older database. It is not really all recent one. And they are mostly talking about antibiotic productions. One group of the enzymes producer aspergillus. This is of course not of course microorganism, but not definitely bacterial species. We can also have some fungal species for producing different kinds of enzyme, and our objective use of the molecular biology tools can be used. These things I was telling today, cloning of genes, coding for enzymes, that can be user for inhibitor. Some reporter assay we can do. Molecular probes, I was telling, we can have the sequence. If I know, I can put this and I can select this, identify particular group of microorganisms. So these tools, cloning of genes, okay, or we can have some provision for test organism known for some agents. If I know some sensitivity of the microorganism, reporter genes you can find is, for example, Lac Operons. Okay, so Lac Operons are a group of genes, cluster of the genes, which can actually utilized by microorganism to feed on lactose. So if in the media, if lactose is there, the sum of the genes will be expressed. They will be producing some protein for utilizing the lactose. So these kinds of operant system, these, I think these terminology you don't know right now. So I'm just little skeptical whether you all understand or not. Some substrate can actually activate some gene. So if it is present lactose, beta galactosidase gene will be activated. Otherwise, it will not be activated, because only need based. If it is required, then they will produce. And these can be used as a reporter genes. So these genes can be actually manipulated. They can be inserted to know whether our target gene is present there or not. We can identify that. So molecular probes, yeah, these are blotting techniques. In molecular biology, possibly you will be studying more details, but basically they are looking for some specific proteins, specific gene sequence involved there. Based on that, we are having northern blotting and southern blotting techniques are there. And we will discuss a little bit more in our upcoming days, upcoming class, these molecular biology techniques more, not from the text, possibly some outside of the text. Otherwise, it is not there here. Anyway, I'm stopping this discussion here today. Something more we'll be discussing in our next class. If you have any queries, you may please raise. You are very silent.